My name is Candace Galen, and I'm a professor in biological sciences at the University of Missouri-Columbia. The slide that we're looking at is a view of our field site at about 3,700 meters, so that's roughly around 12,000 feet. You can see Polemonium viscosum. It has very showy purple to blue flowers. For decades now, we've studied plant relationships with their animal pollinators. Well, we're very interested in how they communicate with one another. How is it that a bumblebee knows where a rewarding flower is? What signals is that plant sending to the bee that the bee can perceive? And so part of that is work that we've done with floral fragrances. And to ask that question about fragrances or volatile signals, we need to take essentially electronic noses out into the high alpine with us, and those are simplified fragrance collectors that we can then take apart when we get back to the lab and get a sense of the key compounds that the bee might be finding attractive, or in some cases, as our work has shown, repellent, and the concentrations of these sort of compound. We've noticed that the fragrance of Polemonium viscosum, the alpine sky pilot, while the flowers of this species have more than 20 volatiles that they produce, there's one in particular called 2-phenylethanol, which accounts for about 95% of that fragrance stream. This species of bumblebee is Bombus balteatus. It is the biggest bumblebee that we have in the Colorado Rocky Mountains at these high elevations, and it has the longest tongue, which is important ecologically because the longer the tongue of the pollinator, the longer the tube of the flower, and they're closely matched to provide the pollinator with a tongue long enough to retrieve the nectar, and the flower long enough to force the pollinator into the flower to do the pollinating. And that becomes important because the 2-phenylethanol, as I mentioned, is at the base of the flower. And so the bumblebee is able to slurp that up without sticking her head in where she would be breathing in great amounts of the 2-phenylethanol. Now imagine an ant, the ant in particular that we're looking at, Formica neorufibarbus gelida. So when the ant crawls into the flower and crawls down to the base, it's completely surrounded by whatever fragrance that flower is producing. The ant is, because of its size and its behavior, getting a much different dosage, if you will, than the bumblebee is. Now, that becomes important because 2-phenylethanol, in fact, is a volatile that has a deleterious or a negative effect, a defensive effect in these flowers. So it's very interesting that the volatile compound, which accounts for 95% of the flower fragrance, is not an attractant at all. So that this fragrance compound has a defensive role, and it actually is highly repellent to ants and somewhat less repellent to bumblebees. And we find that the plants that ants visit produce very little 2-phenylethanol. The plants that they avoid produce a lot. Now, why wouldn't all plants produce a lot of 2-phenylethanol? Bees also find this compound repellent at high concentrations, but they'll tolerate it at lower concentrations. What happens at lower concentrations of 2-phenylethanol for the bees, which is very interesting, is that they'll drink less of the nectar than they will if you just give them sucrose solution that has no 2-phenylethanol. So 2-phenylethanol is a way to be what we call an ecological sanction. If a bee is too greedy, then it'll get a stronger dose of the 2-phenylethanol because it will remain in the flower and exposed to the volatiles that are released from the base. One of the wonderful strengths of the university to an ecologist is that diversity that we see in an ecological web is mirrored in the diversity of faculty expertise, graduate student expertise, and even undergraduate passion for research experiences here at MU.